At Bioaster, we developed a platform for the generation of artificial scaffold proteins called binders that specifically recognize a target of interest. This artificial binder generation platform is an alternative to the generation of monoclonal antibodies. In contrast to monoclonal antibodies, protein scaffolds are stable, small, and lack post-translational modification. Our platform is based on a combination of ribosome display, next-generation sequencing, and bioinformatic analysis. This combination enables identification of binders with high affinity towards a specific target. Our platform combines 5 steps, starting from a protein scaffold depending on its 3D structure. A library is designed in silico with randomization of the variants at defined positions at the binding surface. We can obtain 10 to the 11th to 10 to the 15th variants per library. The scaffold library is used for self-reproduction. Using ribosome displome approach, after transcription and translation, a ternary complex is generated containing RNA, ribosomes, and the nascent proteins. The resulting nascent proteins are then screened against the target of interest, either in solution, on solid phases, or with target competition. The DNA sequences are obtained after RNA recovery and RT-PCR. The DNA can then undergo a new cycle. After repetition of 3 to 4 selection cycles, the DNA is sequenced. Sequences were translated and aligned. A bioinformatics script has been developed at Bioaster to detect enrichment of specific variants. This script enables sequence extraction at hypervariable positions, calculates individual variant abundance, generates motif extraction, and therefore facilitates binder sequence identification. The identified binder sequences are cloned for E. coli transformation, and recombinant binders are produced at 1 liter scale prior to purification. Target binder affinity is determined by biolayer interferometry. Specificity assays are determined by ELISA or alpha ELISA assays. Stability of binder-target interactions can be addressed by circular dichroism. Binding site interaction studies can be assessed by epitope binning or epitope mapping. The workflow for artificial binder generation combines these 5 steps. Using ribosome display technology, specific binder generation can be fast, 2 to 3 months, without need of immunization, and results in binder generation with high target affinity, around nanomolar range. The production process in E. coli is reproducible, low-cost, and GMP compatible. The specific bioinformatics script creates reproducible and scalable data analysis that facilitates binder sequence identification. The combination of ribosome display, next-generation sequencing, and bioinformatic analysis provides us a powerful protein evolution method for reproducible and robust binder generation with improved affinity, specificity, and enhanced diversity landscape. Today we have a catalog of 4 scaffold libraries that allow to select the most suitable scaffold library that fits with the biological needs. There are 3 main applications. The first is the generation of ligands for diagnostic reagents, for in-process control or in-process monitoring or release tests. The second is the use as biotherapeutics against protein targets that are relevant disease-related molecules. A strong increase of clinical phase trials have been noted in the last 5 years. The third application is the use of binders for research use to get better understanding about protein-protein interactions. Therefore, we are interested in setting up collaborative projects with industrial and academic partners for specific binder generations within these various applications.